Our first speaker is Sanim Rahman. So Sanim has started his academic journey back in South Florida, where he did his BSc, and then he moved for his PhD to Johns Hopkins and worked in Cynthia Wahlberger's lab, which will be the main topic of what he will talk about today. And now he's a postdoc at Roger Greenberg's lab in University of Pennsylvania. And we are excited to have him here and have his talk about USB21 autoinhibition and histone deubiquitination. Sanim, welcome, and the floor is yours.
Awesome. Thank you so much. So I first want to thank the organizers for giving the opportunity to present this work. This will be on a project that I did, finishing up my PhD in Cynthia Wahlberger's lab on this histone deubiquitinating enzyme called USP21. So I hope this talk will be interesting not only for all of us in the fragile nucleosome community, but also those of you in the crowd that may be ubiquitin aficionados, as well as protein structure prediction enthusiasts. So of course, I don't think I need to give much of an introduction. Oh, let me see what's going on with my slides. Okay, there we go. So I don't think I have to give much of an introduction on histones and nucleosomes to you all in the community, but I think we can all appreciate the fact that histone proteins are not just scaffolds of chromatin architecture, but they are diverse signaling hubs. And this is accomplished by the fact that these histone proteins can undergo a vast variety of post-translational modifications. And the ability of these modifications to regulate distinct biological pathways depend on not only which histone residues deposited on these, which modifications deposited on these residues, but also which histone residue is the acceptor of these modifications. And in the Wahlberger lab, one of the favorite examples that we have is the ubiquitination of these histone proteins. So in the context of the ubiquitination of histone H2B lysine 120, this is a post-translational modification associated with transcription activation or new chromatin formation. And on the flip side, the ubiquitination of histone H2A lysine 119 is a modification associated with heterochromatin compaction and transcription silencing. And so, of course, transcription is not stagnant. Our genes are not always stuck in a state of on or off state. They are dynamically regulated. And likewise, so is these histone modifications as well. And in the context of H2AK119 ubiquitin, this modification can be removed by deubiquitinating enzymes, which we'll be abbreviating as DUBs. So in vertebrates, we know of 4 major H2AK119 DUBs, and that is USP21, USP16, the PR DUB complex, also known as the BAP1 ASXL1 heterodimer, and MYSM1. And so we know that these 4 DUBs target H2AK119 as specific foci, but really the mechanism of action wasn't really well understood the past 20 years. I would say a lot of progress started to come in the last 2 years, which I'll touch on a little bit more. But particularly, the focus of my talk is on USP21, which I will say is actually the least studied H2AK119 DUB out of these 4. And so USP21 is not just the H2AK119 DUB, it is also responsible for disassembling ubiquitin chains, particularly K48 and K63 ubiquitin chains on a variety of cytosolic and nuclear proteins. And so therefore, USP21 overexpression and mutation has been found to be implicated in various cancers. And so as a structural biologist, I was quite interested in understanding, well, how does USP21 engage with H2AK119 ubiquitinated nucleosome? And biochemically and structurally, is there anything we can learn to understand its general principles and how it removes ubiquitin off substrates? And so if you look at USP21, from a bird's eye view, just very basically at its domain architecture, you'll see that it's really not that complicated of an enzyme. So at the C-terminal end of this enzyme, you have this catalytic domain. So this is the ubiquitin-specific protease domain. This is a structurally conserved catalytic domain shared by over 50 plus DUBs in the USP family. And then preceding this catalytic domain, we have this intrinsically disordered region that's about 200 residues. which we don't have much of an understanding of how it functions, other than the fact that it houses a nuclear export sequence and has been reported to be involved in some substrate binding. So for my initial work, I really focused on this catalytic domain of USP21 and understanding how this portion of the protein engages with H2AK119 ubiquitin. And so I've resolved the cryo-EM structure of the USP21 catalytic domain bound to an H2AK119 ubiquinated nucleosome. And so you can see that I'm able to clearly discern the densities of USP21 in cyan and ubiquitin in magenta. And you can see that it straddles onto the surface of the histone octamer. And so from this structure, we're able to identify some key molecular features for how USP21 specifically recognizes H2AK119 ubiquitin. And so one of the first features that we were able to identify was that the H2A C-terminal tail remodels itself in order to orient ubiquitin into the catalytic site of USP21. So if you look at a crystal structure of an unmodified nucleosome, you will see that the C-terminal tail of histone H2A, where ubiquitin is conjugated to, lays flat on the surface of the histone octamer. But when ubiquitin is conjugated to this tail and USP21 is bound to it, we see a subtle conformational shift where the C-terminal tail lifts itself up off the surface of the histone octamer, orienting itself closer to this catalytic site of USP21. Along with this repositioning of the C-terminal tail of histone H2A, we identify two key interaction interfaces between USP21 and the nucleosome. The first of which really isn't much of a surprise to us is the nucleosome acidic patch, which, to remind you all, is a cluster of negatively charged amino acids contributing by H2A and H2B. And we know from some fantastic work from Rob McGinty's lab that about 50% of nucleosome binding proteins utilize this region for nucleosome recognition. And to no surprise, USP21 also utilizes this region for nucleosome binding, where it utilizes a canonical arginine anchor to bind to the acidic patch along with this neighboring lysine residue. And so I don't have time to show all the biochemistry data, but you can check our preprint and forthcoming publication on this work where we biochemically validate these residues and others I will show important for this interaction interface. And along with this acidic patch, on the back end of USP21, there are two arginines that position itself to engage with the nucleosome DNA, particularly at superhelical location 6.5 by binding to the negatively charged phosphate backbone of the nucleosome DNA. And so I don't show these features just for the general appreciation of USP21 structure-function relationship. I would say that these results can be best appreciated in the context of work done in the last 2 years, which are from fantastic works from Mingming Xu, KJR Mosh, and Lei Lu's labs, who have solved the structures of the PR-dub complex in USP16 bound to an H2AK119 ubiquinated nucleosome. And I'm showing you here a side-by-side comparison of our structure and their structures as well, showing how all three of these dubs engage H2AK119 ubiquitin. And what I hope you would appreciate just from first glance is that all three dubs have very divergent modes of nucleosome recognition. You can see that not all of these rely on the acidic patch for binding. They all have similar binding modes to the nucleosome DNA. The only thing that they have in common is that they make contact points with superhelical location 6.5. And then you can see that they all drastically reposition ubiquitin across all three. So this was quite a surprising finding, especially with USP16 and USP21, where we find that the actual sequences differences between the two is what caused this divergent mode in binding. And so I would say that about right now, we're halfway through this talk. And appropriately, I have only so far have talked about half of this protein itself, which is just the catalytic domain. And so if you recall from a few slides ago, I've talked about that there's also this intrinsically disordered region on USP21, which really we can't address by cryo-EM. And so I became quite interested in this disordered region to see, well, how does it contribute to USP21 function? So what I did was I expressed and purified the full-length USP21 from E. coli and a series of N-terminal truncations as well, and assessed its biochemical activity when you have USP21 in the presence of this intrinsically disordered region. So I carried out first a set of activity assays on K48 ubiquitin chains, and I ended up finding something quite interesting. So I found that the full-length USP21 essentially had little to no activity on these K48 ubiquitin chains. But as I started making up a series of N-terminal truncations onto this disordered region, you can start to see that I am able to gradually restore the activity of this enzyme, the highest being just the catalytic domain of USP21. And so in our paper, we showed these on a series of other substrates, but most importantly, we also showed this on H2A-K119 ubiquitinated nucleosome as well, where you can see by EMSA, you can see that the full-length enzyme is unable to effectively bind to the nucleosome core particle, and it also has much poorer activity on nucleosomes as well and cleaving ubiquitin from it. And I just want to give a quick shout out to the sponsor EpiCypher, who creates a fantastic ubiquitinated nucleosome product line. These are extremely difficult to make native linkage ubiquitinated histone, and I highly, highly recommend giving them a try if you're interested in looking at histone ubiquitination. These are extremely difficult to make, and these products are always been very high quality in our hands. So big shout out to them. But going back to USP21, so from these results and others, it became quite clear that what we have in our hands is potentially that this disordered region is an autoinhibitory module of USP21. And so the concept of autoinhibition in deubiquitinating enzymes is really not a foreign concept whatsoever. In fact, a lot of dubs in eukaryotes are actually exist in an autoinhibited state, and they're activated in by different mechanisms. And there's three main mechanisms for how autoinhibited dubs are relieved. So the first of which is that some of these dubs are autoinhibited due to oligomerization, some that are activated by post-translational modifications. Largely, we know many of them are activated by phosphorylation. And then a good number of them are also activated upon specific protein-protein interactions or in the formation of specific complexes. So we know that from biochemical studies, solution studies specifically, that it couldn't be oligomerization because the full-length USP21 and all its N-terminal truncations always existed as in a monomeric state in solution. And so I know it had to deal with either with something with post-translational modifications or partner proteins. But I really had no idea what avenue to take. And so what I speculated was that if I wanted to know which one of these doors to walk through, I needed to know what are the interactors of the disordered region, of USP21's disordered region. And then I can maybe gain some hypothesis on how could autoinhibition of USP21, how does this autoinhibition get relieved? And so I would say that I got really inspired by some fantastic work done in the last few years using AlphaFold, not in the way that you would think for general protein structure prediction, but as a tool for in silico protein-protein interaction screening. So particularly I want to give a nod to Johannes Walter's lab and work that he has done with Lucas Farnung, where they used AlphaFold to do in silico protein-protein interaction screens to look at the interactomes of nuclear proteins. And in this case, for this paper, looking at interactors of the nucleosome acidic patch. And so I thought this was a very clever way to look at the structural interactome of proteins. And I thought I could also use a similar approach to this to identify interactors of USP21's disordered region. And so I developed my own modified pipeline to see if I could identify interactors of USP21 using AlphaFold. And so what I did was I extracted the 152 known interactors of USP21 from this interactome database called BioGrid. And I co-folded all of these 152 interactors with both the catalytic domain of USP21 and also the full-length USP21. And what I rationalized is that I could extract the interface predicted template modeling score. This is also known as the IPTM score, which is AlphaFold's confidence in the structural interfaces of two or more models in two or more proteins in a model. And what I rationalized was that if a protein is an interactor of USP21's IDR, I would see that it would have a higher IPTM score with the full-length USP21 over the catalytic domain. And so out of the 152 interactors that I screened, I found that only 13% of proteins of USP21 interactors had an improved IPTM score with the full-length USP21 over just the catalytic domain alone. And so I'll walk you through what this data looks like. So here I am showing you is each point represents one of the 152 interactors that I have screened by AlphaFold. And I'm plotting them against the IPTM scores when they're predicted with the full-length USP21 on the x-axis, and then on the y-axis against just the catalytic domain. And I want you to pay attention to the points that are more in blue, which are interactors that have a higher IPTM score with the full-length enzyme. And what I ended up finding from the screen was that a large number of proteins that had a higher IPTM score with the full-length USP21 are either kinases or they're phosphoserine binding proteins, specifically these class of 1433 proteins. And what really makes this finding really interesting is that if we take a closer look at these models, particularly at these kinases and 1433 proteins, you will see that in the case for these kinases, MARC1 and PRKCI, their active site residues are poised as if it's ready to phosphorylate one of two serine residues on USP21's IDR, serine 115 or serine 113. And in the case for these 1433 proteins, their phosphoserine binding pocket is poised to bind to serine 113 or 115 as if it was a phosphorylated serine. So then this really made me think, well, potentially autoinhibition is relieved by phosphorylation. But from a structure function perspective, it still didn't make sense. Why would phosphorylation of these residues relieve autoinhibition? And really, I had this answer in front of my face the whole time, but I never really recognized it until I did this AlphaFold screen. Oh, sorry. I forgot to also mention that what made these serines residues specifically interesting is that they have also been identified to be phosphorylated over 90% of the time from mass spec data when you look at these in HeLa cell lines as well as HEK cell lines as well. But going back to the mechanism of autoinhibition, what I had the answer in front of me of how this IDR autoinhibits USP21 this entire time, but I never really recognized it until this screen. And so if you look at the AlphaFold model of full-length USP21, and so in cyan, I'm coloring the IDR region. You can see that, to no surprise, it's complete spaghetti as you would expect. But you will see that a part of this IDR actually folds in onto the catalytic domain and occupies the ubiquitin binding interface, which I'm showing you in purple. And what you will see is that the serine residues that get phosphorylated is what makes this interaction interface between the IDR and the catalytic domain of USP21, particularly serine 113 and 115 sticks itself in on the ubiquitin binding interface. And if you look at the predicted aligned error, you can see that this interaction interface is predicted with high spatial confidence as well. So then I thought maybe this IDR is serving as a mechanism of sterically blocking ubiquitin binding and could be relieved by phosphorylation. And so with AlphaFold 3, what I did was I created a series of USP21 models with different serine phosphorylated serine combinations using well-known phosphorylation sites on USP21. And so I found that if you phosphorylate serine 93, 113, and 115, this IDR no longer occupies the ubiquitin binding interface of USP21's catalytic domain. So from this, it seemed like maybe this IDR is autoinhibiting USP21 by blocking ubiquitin binding, and then phosphorylation relieves this. Is this true experimentally? And so if I do activity assays of recombinant USP21 and treat it with either the MARC1 or PRKCI kinases, you can see that I can increase the activity of these enzymes of USP21. The highest being if you treat USP21 with both kinases, with MARC1 and PRKCI. But the highest activity that I was able to see is if I create a triple alanine mutant at serine 93, 113, and 115, where I'm able to nullify the hydrogen bonding between the IDR and the catalytic domain. And this gives it the most robust activity in activating the enzyme. And so this is true not only for this fluorescent ubiquitin substrate that we use to look at DUB activity, This was also true for H2AK119 ubiquitinated in nucleosome as well, importantly, where the phosphorylation of full-length USP21 increases its activity and allows it to effectively deubiquitinate H2AK119 ubiquitin. And so with that, I'll jump to my conclusions, which is I hope I was able to tell you a very interesting story about the ordered and disordered of USP21 and how by cryo-EM we're able to show that USP21 utilizes a unique binding mode onto the H2AK119 ubiquitinated nucleosome in comparison to other DUBs that have been reported. And that with this N-terminal region of USP21, it functions as a autoinhibitory module. And then by using AlphaFold, in silico protein-protein interaction screening and guided biochemistry provide a model for autoinhibition and relief. And so with that, I would like to thank the Wolberger lab, especially Cynthia, for giving me the freedom to work on this project, and then lab members Chad Hicks and Alex Guizdala, who contributed to this project, our funding sources. And then this paper is currently on preprint, but will actually come out at the end of this week. If you are interested in reading more, there'll be updated data compared to the preprint that I did not have time to show today. And so with that, I'll be happy to take any questions.
Thank you, Sanim, for this great talk. The audience, if you have any question, you can raise your hand and I will unmute you, or you can write in the Q&A or chat box if you want. Ben, do you want? There's answer question. You had a question.
Yeah, that was a really nice presentation. I may have missed this, but is it known how the phosphorylation is regulated? And is this like a toggle to regulate its DUB activity?
Yes, that's a great question. So the one thing that we did not have time to fully address was understanding the mechanism for when phosphorylation occurs. And actually, it's very important that I acknowledge that we are not claiming like MARC1 and PRKCI are the bona fide kinases for activating USP21. Our major point was that these residues on USP21 is what activates it. The next question is, of course, as you ask, yes, when are these residues phosphorylated, and what are the actual true kinases important for H2AK119 specific activity?
Yeah, it's very. I mean, I'll be excited to see how this line of analysis kind of extends and what we find out next.
Yeah.
So the first question and the Q&A is from Maria, which asks, beautiful presentation, real cool stories. Curious if you had any tips on how to perform a similar AlphaFold screen for interaction partners. How easy was it to adapt the method? Any unexpected challenges?
Yes. So there is a pipeline that I use, and I cite it in the preprint and paper called Alpha Pull Down. So this is a Python-based package where you can automatically do the screening. So I will say that this approach I did, I would say is a bit outdated. There's better ways to now score interactors. I recommend, highly recommend checking out this preprint by Roland Dunbrack at the Fox Chase Center on this score he developed called IPSAE, which I think is the best way to identify bona fide interactors using AlphaFold. So I think it's quite possible if you want to use, if you have a workstation set up with AlphaFold, it's quite possible. Or you can even use the AlphaFold 3 server and do it brute force method way like that, where you can get 30 predictions per day for free. And then just use those to and use the confidence scores from there to analyze interactors. So both are possible, whether you have a workstation or not. You can use the AlphaFold 3 server. But I highly recommend understanding some of the confidence scores metrics and utilizing this IPSAE score that I talked about.
Thanks, Sanim. This next question is from Genevieve asking, did you try to mutate S to E instead of A?
Yes, so we did try an S to E mutation. We end up not finding that it had any effect in activating the enzyme, which is which was quite puzzling for us. But then when we talked to others in the field that have worked on DUB autoinhibition, they have found that these phosphomimetics, particularly the serine to aspartates or glutamates, actually only about 20% of the time do they actually work. So I think this was a comprehensive screen done, I think back 10 years ago, biochemically looking at the use of phosphomimetics and so on the test cases they found only about 20% of the time did it work, which was quite actually surprisingly low from what I would expect. So we think that maybe the phosphomimetics were not the most effective in this case, but we did try that originally.
Thanks, Sanim. If anybody has more questions, you can write in Q&A box and Sanim can type the answer, or you can contact him later for your questions. So